Thank you, Dr. Ansrud. That's very informative. I look forward to a lot of questions, and we really appreciate some of the very practical, hands-on demonstration, and thank you for Dr. Karam for being a guinea pig today. Our next presenter, we're thrilled to have him travel from the East Coast to join us today, although he'll share from you his personal experience that this is kind of a homecoming for him. Dr. Matt Harms is an assistant professor of neurology at Columbia University in New York City. He has his medical doctorate from University of California, San Francisco. He completed his neuromuscular medicine and clinical neurophysiology fellowships at Washington University in St. Louis. He's past director of the Washington University Neuromuscular Genetics Program. At Columbia, he's continued his work harnessing emerging genetic technologies to understand the cause of inherited neuromuscular disease, including ALS. He leads an international multi-site team using whole genome sequences in an effort to bring precision medicine to ALS. So join me in welcoming Dr. Matt Harms.
Thank you. Yes. So Lance mentioned that this is a bit of a homecoming. I actually grew up in Eugene, so although I'm coming to you today from Columbia, I actually got my start here in Oregon, and just felt my blood pressure lower as I got off the plane and could smell the evergreens, although that might have been the lack of exhaust fumes from New York City taxi cabs. I'm not exactly sure. So today I want to talk about the genomics of ALS. We used to call it the genetics, but that's because we could only study a limited portion of the genome. And now that we can do the entire thing for relatively cheaply, we're starting to call it the genomics of ALS because we're starting to find out about regions of the genome implicated in the disease that we didn't know about previously. And I want to talk today about how we're using that to guide trials and then to generate targets for therapeutic development. So we already heard some of what ALS is, and I just want to remind everyone that it is a complex disorder. It's called a motor neuron disease, but now we're learning that in fact other neurons, including those in the frontal executive circuits at the front of the brain, sometimes those in the deep centers of the brain where the smoothness of movement is controlled, and then even some patients have involvement of what are called the dorsal ganglia, which control sensation. So although the motor neurons are the most deteriorated and the most affected by the disease, other neurons are involved. And within a given patient, there can be any number of combinations of these different regions that are involved, which means that in the clinic, every patient looks different one to another and makes it a challenging disease to study. So who is it that gets ALS? Unfortunately, many of you in the room know firsthand exactly who that is because you may be among them or your loved ones may be. But it turns out that actually this is what I would call a not-so-rare disease. So 1 in 3 per 100,000 will develop the disease in a given year. And that means that living around the world, there are close to 300,000 people with it. In the United States, that translates into about 30,000, or here in the Oregon, Washington, and Idaho region, about 750. So it's not that uncommon. We do know that there are some groups of individuals who are at increased risk. So males have a slightly increased risk. Elite athletes you heard about earlier, those who have served in the military, whether they've seen combat or not. And then those of European ancestry compared to African or Asian ancestry are also at increased risk. To put that in a little bit of context, that means for me, my lifetime risk of dying of ALS is actually 1 in 372. And to put that in reference, it's a little less likely than me dying in gun violence, which is 1 in 280. And a little bit less likely, or more likely, than being struck by a car and dying as a pedestrian. So it's not that uncommon, although I know sometimes it feels like you're all alone in suffering with this disease. So one of the strongest risk factors, of course, for developing ALS is having a strong family history. So when someone comes to my clinic, there's somewhere between a 7 and 10% chance that someone in their family will have had the disease at some point. And if you have a strong family history of ALS, your chance of getting the disease is actually about 1 in 3. And in some families, it's more like 50-50, but overall, it's about 1 in 3. And it's actually from studying those individuals with a very strong family history that we've learned the most about ALS, what causes the disease, by studying the genes that underpin this family history. You saw a very similar slide from Dr. Karam's talk illustrating the gene discovery history in ALS. So 1993, just as I was completing my high school time at North Eugene High School, was when SOD1, the very first gene, was discovered for ALS. And there's been slow and steady progress in the next decade. The human genome was actually published in 2000, and that gave a tremendous boost in the ability to discover genes, made it faster and simpler. But really, it wasn't until 2009 when a new gene analysis technique called next-generation short-read sequencing came online that the real pace picked up. And you can see that basically since then, it's just skyrocketed the number of genes that we know. And up here in this upper region, I'm happy to report that many of the projects were actually funded directly by the ALS Ice Bucket Challenge, something that many of you participated in and that got people very excited about helping out in ALS. So how do gene mutations work? So I thought I would just go through this at a very basic level in case you've forgotten some of this from high school biology. So the central dogma states that the DNA contains the instructions for proteins. And a copy of the DNA first gets turned into a set of code called the RNA, and then the RNA itself goes on to the protein. Because you inherited one chromosome from mom and one from dad, you have two copies of the CHOMP gene, which I've illustrated here, and two copies of the linker gene. Of course, you actually have 19,000 genes, but we'll just focus on these two examples. And so what first happens is a process called transcription is utilized, where the CHOMP DNA set of instructions is copied into the RNA set of instructions. Both both chromosomes will be utilized, so you'll have a copy that came from mom and a copy that came from dad. And then both of those, if they're normal, will get translated into a protein. And so here, the CHOMP protein looks a little bit like Pac-Man because its job is actually to cut whatever this happens to be in half and leave it in pieces. So that's its normal function. Some cells will use the CHOMP protein, others won't. It kind of depends on what type of a cell you are, which of the different genes you're going to use. And so you can imagine another gene, the linker gene, gets used in some other cell types. And so similarly, it gets transcribed into RNA. And then that gets used to manufacture the protein. And in this case, it's the linker protein because it pulls these two other proteins together so that they're close enough to do the job that they have to cooperate to accomplish. So how does a mutation mess this up? So if we now take a look at the CHOMP and linker proteins and introduce an abnormality in the DNA, that abnormality in the DNA will now show up as an abnormal code in the RNA. And then when the translation occurs, the protein is defective. And so now the mouth of the CHOMP protein isn't close enough to actually cleave this rod. And so uncleaved, it just kind of starts to accumulate. And the clumps of the rod basically get in the way of the cell's function. Similarly, for the linker, if there's a mutation located there, this copy of the RNA will be abnormal, and it may lead to an abnormally globular shape. And so although this protein can now grab onto the red one, it can't bring the blue one close enough for them to do their cooperative job. So that's how a gene mutation leads to malfunctioning proteins. And then if that protein is important enough, it goes on to lead to problems in the neurons and cell death. So all of those genes that I showed you on that graph have functions. And so if you start to look at the functions of all the genes we've discovered in ALS, you can start to see some themes that tell you which pathways are really important for keeping motor neurons alive and which pathways are going wrong that lead to ALS. And so if you put them on lists, you can start to see that a process called protein homeostasis is really important, a process called cytoskeletal and axonal transport, where things that are manufactured up here in the cell body have to get all the way out here to the very end where the muscle's located. We now know from these types of analyses that metabolizing the RNA, that middle component that I showed you previously, and then shipping neurotransmitters and other things via endosomes are also important. So one of the things that we've learned from uncovering the genetics of ALS is not just who has carrying the gene, although that's very important, but we've also learned kind of which pathways are super important for keeping motor neurons alive. So how frequent are mutations in these genes? So we did a study just as those next generation sequencing techniques were being released, which allowed us to look at a large number of patients for the very first time. So in the WashU Neuromuscular Genetics Project that was mentioned earlier, we had 42 individuals who had a very strong family history, and 350 people who were so-called sporadic ALS. No one in the family had ever had the disease. And we were able to, because of the cost of sequencing coming down, analyze 17 ALS genes across both of these groups. Previous to next-generation sequencing, it would have cost us about $500,000 to do this study. With the falling costs from next-generation sequencing, we were able to do it for about $25,000. So there was a huge savings. And if I redid this study today, it would probably only cost me $2,500. So the cost of sequencing has dramatically fallen. And what we learned from this study is that we could find a causative gene mutation in the C9, SOD, TDP, these other genes, in about 60% of families. And in sporadic ALS, we were surprised to find those exact same genes and those exact same mutations in 9% of people with sporadic disease. So completely unsuspecting that they were carrying a gene mutation. But that means that there's, I guess I should also point out that we've now gone from analyzing 400 patients to analyzing 5,400, and the numbers still hold true. We're learning new, interesting things by this much larger analysis, but this illustrates just how broadly sequencing can happen now because of the costs coming down. So that still means that in sporadic ALS, we don't find an explanation in 91%, and if you look over in the familial cases, even to this day, we still don't find an answer in about a quarter of individuals. So that means that there must be other genes out there or other things going on. So what else is going on in sporadic ALS? So I've illustrated for you those genes that cause ALS, where one bad mutation is enough to trigger the disease. And so that's one thing that could be happening in that 9 or 10% of sporadic disease. But it's also possible that you could have 3 minor genetic problems that kind of all pile on one to another to trigger degeneration. You could also envision, though we haven't discovered one yet, a massive environmental exposure that could trigger neuron degeneration. Although viruses, some types of viruses are implicated in ALS, the West Nile virus, as soon as you get infected, it kills off motor nerve cells. So you could envision an environmental exposure that was strong enough to cause ALS all by itself, but we don't know of one yet. I want to emphasize that. But you could also envision that there would be multiple smaller environmental exposures. So maybe being an elite athlete and in the military and a male and exposed to X, Y, or Z could all add together to cause disease. But probably in sporadic disease, it's a series of genetic problems plus a series of environmental exposures that eventually go on to cause the motor neuron to degenerate. And the technologies that we have today have allowed us to make lots of progress in identifying one major genetic problem. And we're just now on the cusp of being able to look at multiple genetic problems and this combination. So another way to kind of visualize how this is probably happening is that if this scale represents the health of your motor neurons, and a tilt to the right under the insults category leads to motor neuron degeneration, if you have a really big bad mutation, like the A4B mutation in the SOD1 gene, that is a massive insult. And although your body rallies and tries to compensate by expressing other genes, trying to turn down the SOD1 gene, it's just not enough and could never be enough. And so everyone with the A4B mutation, the balance tilts and they develop the disease. You could also then envision smaller effect mutations. So this would be another mutation in the SOD1 gene called the I113T. And the compensations in about 50% of patients are sufficient to prevent the disease from ever occurring. Yet we know that being male is a risk factor. So now if you add in just having only one X chromosome instead of 2 because you're male, that's enough to tilt. And we know that the SOD1 mutation, I113T, manifests more frequently in men than in women who carry the disease. So now you can also envision taking that one step further to these smaller effect mutations, and these wouldn't necessarily have to be in the same gene. And so you could have 3, 4, 5, who knows, we don't know what the upper limit is, little tiny mutations that minorly affect 3 or 4 different proteins. And those compensations could be quite adequate. And so many people could be walking around with these same risk factors. And then you could add in cigarette smoking or some other little feather of a risk factor that adds in and triggers the disease. So what we think of ALS from a genetic perspective is that it's a complex disease. And so there are situations where one gene mutation is sufficient to cause the disease, and at the other end of the spectrum, a pile-on effect or synergistic effect from lots of different minor genetic problems, plus some environmental risk factors or lifestyle factors, and that most people are somewhere on this spectrum. And if they're at this end, we call it monogenic, or one gene, and there's a single cause. If it's at the other end, we call it multifactorial. And then really we're going to recognize these individuals as familial ALS because those big bad gene mutations are really strongly active. And then this is what we're calling sporadic ALS at the other end. But it's a spectrum and a continuum, and they're all genetically influenced. So while you may have sporadic disease, there are still genetic contributions that are playing an important role in your disease, though they may not be these big bad mutations that you're likely to pass on. So I'll go back to this ALS discovery timeline because there are now, I would say, 20 to 24 causative gene mutations. There are actually 100 claimed. So there are a lot of genes that aren't actually ALS genes that have been said to be. But that raises the question, how many undiscovered ALS genes are there? And there are some complicated statistical models that actually come from ecological estimates of species in forests where you can't go out and count all of them. And it suggests that there are 100 yet to go for us to have identified all of them. So that begs the question, why continue the gene hunt, which is what I do. So there's job security, so I may be a little bit biased, but I think that there are some good scientific reasons. So the first of these is to provide answers to some key questions that are definitely pinging around inside your heads, which is, why me and not someone else? And could this happen to those I love? Could I pass on genetic influences that make them more likely to develop the disease? And that's a very important question and one that I answer every day in clinic as we do these genetic tests. More importantly, I think kind of to ALS in general, is that we want to be able to empower what we call precision medicine. So the idea for precision medicine is that we're giving the right drug to the right patient at the right dose at the right time. And so we want to be able to use genetics to help power the next generation of clinical trials, to identify responder groups for drugs that we already have. It could be the reason why Riluzole doesn't look like it helps that much is because we've given it to everyone, and that there's a subset who do really, really well on it and a subset who do really poorly, and we could be able to distinguish that. We also want to be able to use genetics to guide the best choices among drugs once we have a whole host of ones to choose from. And then I think most importantly, we really want to identify new targets, things that might be easily druggable and fixable from a therapeutic perspective. And that could include the individual proteins that we learn about and then important cellular pathways. So how do we do that? So Shafiq already alluded to the fact that we've now had 60, 70 randomized clinical trials that have led to 2 approvals. And what should we have discovered if we were like the other diseases that the pharmaceutical industry has targeted? So typically 10% of drugs in trial make it to FDA approval. So by my calculation, that means that we should have approximately 6 by now, and we only have 2. So what's been the problem? Well, one of the problems goes back to this snapshot here, and that's that we take all comers in our ALS clinical trials. If you meet the clinical diagnostic criteria and in the inclusion and exclusion criteria, you make it into the clinical trial, and that basically assumes that ALS is one disease, that everybody coming to the clinic, everybody joining the trial, has the exact same underlying mechanism, and I think the genetics is telling us that that's not the case. So some of the problems with the ALS clinical trials that have happened in the past not proving that the drugs work, some people call them failed trials. I like to call them negative trials because I think we did learn from them, and failure implies that it was just a waste of our time, and that's far from the truth. So I think we learned that there are some drug-specific issues in the way we used to do studies. Number one, we didn't realize that most of the drugs we were trying didn't get across the blood-brain barrier and actually get to the neurons. They were in the bloodstream, they were in the liver, but didn't make it to the brain. So obviously had 0 chance of saving neurons. We also learned a lot about study design issues. We were choosing too small a cohort. We were doing the studies for too short a period of time, and we were shooting for the moon and anticipating a much larger effect size than I think we've learned to expect. But I think the major reason is because we've been ignoring the big difference in underlying causes amongst the different patients that are coming to clinical trials, and that in the past we've been limited in how we can look for targets for drug development. We really only had the SOD1 mouse model, and we really had no way to look back in human tissues to find out whether the genes we were studying were important in the disease or not. So we've made a lot of progress. We now think about this routinely at the very, very beginning of thinking about a drug for clinical trials. The very first thing that gets tested is, can it cross the blood-brain barrier? How long does it reside in the system? Things like that. We now have very sophisticated design trials that morph from phase 1 into phase 2 and take some of these issues into consideration. And so what I'll talk about the rest of the time is kind of how genetics is playing a big role in these last two components. So again, here's the typical ALS clinical trial. They meet the clinical diagnostic criteria for ALS and come into the trial. But we really know that if we could look at the underlying biological cause, we would start to see differences in different subgroups of patients. And so basically, if we could figure out a way to find the individuals with the same underlying causes and parse them into these different groups, then we could perform genetic stratification. This would mean that the participants in a given clinical trial would be more similar one to another and would make the trial easier to perform. It means that we could then use smaller numbers in the clinical trial and potentially even lower the number of people receiving placebo. This would translate into much cheaper trials that could happen more quickly and give us a better chance to see the effect, especially if we get to the point where we're able to pair one of these individual subgroups to the type of drug mechanism that's being proposed. So if you knew that this blue group had a problem with pathway X and the drug you were developing seemed to target pathway X, you could pair up just those individuals with that particular drug study. So that's what we're moving towards in the future by doing these genetic investigations, so that we could take this cohort of ALS patients that all look the same on the outside and figure out who they are and their underlying biological cause. And then figure out, is there a group that's basically neuroinflammation or cellular transport, or what are the genetics and the features of these different groups telling us is going wrong, so that then we would be able to target the right drug that affects each of those different pathways at the right time. And so that's the future, the promise of precision medicine for ALS. And I think at the moment, we're already doing this in some of the strongly genetic subgroups. We would also anticipate that we would find people who didn't fit into these nice, neat categories. And these are just hypothetical categories, by the way. This isn't known yet. So this individual might not have the features of any single group, but might look a little bit like these two. And then we would say, well, let's just try kind of a combination drug where we give them these two and see if that works better than one by itself. So already this is happening for strongly genetic subtypes. Dr. Karam already told you about the anti-SOD1 antisense oligonucleotide. At the American Academy of Neurology meeting, the initial results were released, and in fact, the antisense oligonucleotide does its job. It gets across the blood-brain barrier, and it destroys the SOD1 protein. So that's very successful. And not only that, especially in the individuals with that A4V mutation, that big giant weight that sat down on the scale and tipped it all by itself, the rate of progression was arrested in some patients and slowed dramatically in others, which is not ever heard of in that particular mutation. So this is extremely exciting. And a similar molecule for a different gene, the C9orf72 gene, is also underway. And I needed to update this. I don't know where your nearby sites are, but there are some here. San Diego. Okay, great. All right. So going back to this slide, kind of showing the different pathways that are involved, eventually we'll probably have antisense oligos or similar types of drugs that target many of these different genetic mutations. And I would also argue that the more genes we discover are going to teach us what neurons need for health. They're going to show us some unsuspected pathways that aren't currently on this list. And once we have 15 pathways on the list, then we're going to have the opposite problem, and we're going to need to know which pathways are the most key ones for us to target, because we don't have enough resources to basically design drugs for all of them. Which ones should we zero in on and focus our energies on? So how are we going about identifying more genes? How can we get those 100 that are still hiding out there? And so that's what I'll talk about basically for the rest of my talk. So mainly the way we're doing this is by a fancy statistical test called burden testing. And I promise all of you can understand exactly how this happens. Okay? So if you have a group of controls and you sequence their whole genome, and here I'm just showing one part of one gene, and then you ask in these controls, people who don't have ALS, you ask the computer to show you all of the regions that are different from what we expected from what we call the reference genome, the normal genome. And then you see that in each of these different locations where the stars and lightning bolts are located, there are differences in each of these individuals. Then you ask the computer to predict which of those changes are likely to disrupt the protein, to like prevent the linker from bringing those two things together or prevent Chomper from cutting the rod, and those that are predicted to do nothing to the protein, to not affect it at all. And so you ask it to classify each of the different changes. And then you do the same thing for a large number of cases over there on the left-hand side and ask the computer to do the same thing. And now you basically compare. Are there more deleterious mutations in the patients who have ALS compared to the people who don't in any genes across the genome? So no one knew whether this would work back in 2015 when we did our first study. And so we assembled 3,000 ALS cases and 6,000 controls using this big consortium. And using generous funding from Biogen IDEC, at that time, about $5 million, we sequenced what's called the exome. So that's the region of the genome that makes proteins. It's about 2% of the DNA in our bodies. And what we were able to do is rediscover SOD1 and TDP43, genes that we already knew about. So we knew the technique was working. But we also were able to identify TBK1 and NEK1 as two new ALS genes. And since then, other groups have gone on to use similar methodologies to find new genes for ALS. And we've gone on beyond that now, because we've got to get more sophisticated in how we do these analyses. And we now use something called regional collapsing, where instead of just asking which genes are affected, we're asking like microdomains, like this little tiny region of the gene. Show me the little tiny regions of the genes where ALS patients have mutations more than the cases. And I won't go into the details of this, but these are the different ways that we do it. And we're very excited to report that this also works. And so here's our most recent analysis, and LG-ALSL is the newest ALS gene. You heard it here first. It just came out last week in Genome Research, and it is actually telling us about an entirely unsuspected pathway, just as I was alluding to the importance of on the previous slide. So this actually has nothing to do with any of the other known ALS genes. It takes sugar moieties, called galectins, and it attaches them to proteins that have already been manufactured, and that pattern of galactonization tells the protein where to go. And so it's probably helping to act like the FedEx scanner to like ship proteins to where they need to go and get them to where they need to be delivered. And that's messed up, and we didn't know that that was happening in ALS until the discovery of this particular gene. So souping up the statistical analyses and the way that we look for these new genes is one approach. But I can tell you that the best way to discover new genes is to study more patients. And so really, more patients being sequenced and then numbers is exactly the way of the future and what needs to happen. And so I'm really happy that we've received funding from the ALS Association, as well as from Biogen, to create this study called Genomic Translation for ALS Care. And this is, when we first started the study 3 years ago, this title was somewhat aspirational. We thought, well, you know, someday we'll be able to translate the genome into care recommendations for patients. And I've already told you that for SOD1 and for C9orf72, that's on the horizon, if not already here. So it's no longer aspirational for some component of the population, and hopefully over time will become more and more the norm. So how does the GTAC study work? So basically, here are the participating centers. They're distributed across the United States, and then the entire country or region, however you want to think of it, of Scotland. So patients give us lots of blood. I'm sure that's not new to most patients in most ALS clinics, constantly being stuck and drawn. The DNA heads off to my laboratory. The RNA comes to me as well. We take future stem cells, so blood mononuclear cells that we can turn into stem cells in the future, and we put them in a repository at Cedars-Sinai. It's kind of a library of cells that we can go back to and create stem cells and study in the laboratory. And then a couple of basic lab tests that happen locally to kind of see where people are on some key features. Every 3 months at regular clinic visits or at research visits, we collect data from patients, including the exposures and epidemiological data that is almost identical to the one already being utilized by the CDC National Registry. So that as discoveries and findings come from the CDC, we can immediately turn back to our patients and look to see if there are environmental and gene interactions. And then Unique to our study at the time when we started it was that we wanted patients who are participants to get the information back. So it used to be that when you did a research study like this, if you found something genetically, you weren't allowed to tell the individual. And I said, that's kind of ridiculous. I wasn't alone. There were other people who were starting to say things like that. And so we actually double check every abnormality that we find that we think is causing the disease and use a genetic counselor to return that information back to participants and their families so that they can use that in their family planning and just to have as knowledge. So how's the study going? I need to update this slide as well because we just crossed our 1,000th patient mark. So this has been the fastest enrolling ALS study in the history of the world. And we're pretty proud of that. And that has taken a lot of hard work at places like OHSU. And I'm grateful to their staff who are here who have done a great job of recruiting patients from this region. And we've also made sure that we're working together with people who are doing similar studies in other places. And so in particular, Answer ALS and Project Mine have very similar platforms and similar ideas. And we're all working together because we realize that the more patients we've sequenced, the more genes we're going to discover. And we have to work together in order to get the most out of this. So I would say, and would wrap up today by saying that I think that there are many reasons for hope in 2019 for patients with ALS. So number one of the reasons, I guess I kind of have these out of order, I would say, is that researchers are working together as never before. And part of that comes from the ALS Ice Bucket Challenge money, right? As soon as scientists see blood in the water and some potential funding, they come around, come swimming. And the ALS Association very wisely set up almost every dollar going out in research funding as collaborative work that basically brought people who used to be in their own silos working on their own together. And so now researchers are working together as never before. And the outpouring of that has been molecular therapies that are now nearing clinical use, particularly for SOD1, C9. And I know personally of 3 or 4 genetic gene replacement and knockdown studies that are about to get started. And then finally, new genes are emerging every few months, and I think that's telling us really valuable information about the pathways that are involved, as well as the individual targets that drug companies can go after. And so I was just at a conference for Target ALS, another group that funds research. And when that group started in 2009, there were 5 drugs in clinical development for ALS. And at this particular meeting, we heard about an additional 50 that are in the process. So there's been a 10-fold increase in the number of drugs that are being tested in animal models, cell models, and in humans. And hopefully we'll be able to use the emerging genetic information to take that ratio, 2 drugs out of 60 that we've tried, and increase it to where it should be more on the order of 6 to 10. And who knows, maybe we'll get lucky and even do better than the average disease. So with that, I'll go ahead and pause and hand it over.